In last lecture, we have covered the classification of microorganisms, morphology of bacteria, and bacterial cell wall. And in cell wall appendages, we have covered up till capsule. Okay. So this is the classification of prokaryotes and eukaryotes. Size of bacteria, major shape and their arrangement of cocci and bacilli, actinomycetes, spirochetes. Bacterial anatomy, we have covered the cell wall, plasma membrane, cytoplasm, nucleus, and capsule. Okay, functions of the cell wall, structure of the cell wall, what is the basic difference between the cell wall of gram positive and gram negative organisms. Then cytoplasmic membrane, cytoplasm and its organelles, nucleus, capsule, and the demonstration method of capsule. So the another important appendage of cell wall of bacteria is the flagella. Now here in the picture you can see the long slender thread-like structure that is flagella. So appendage protruding from the bacterial cell wall. Its length varies from 3 to 20 micrometer and diameter varies from 0.01 to 0.03 micrometer. And it is made up of the flagellin subunit. Okay, so this protein subunits, flagellins, they assemble and they form a cylindrical structure with with the hollow core. Now, flagella consists of three parts. One is the long filament, the other part is the hook, and the last and most important is the basal body. Now, in the picture, this you can see the filament portion, long filament portion, which lies external to the cell surface. The hook structure at the end of the filament, to which the hook is anchored with the series of rings that drives the flagella and imparts motion to the flagellum. Here you can see the L ring, P ring, S and M ring. They are responsible for the motion of the flagellum. Now here you can see the macroscopic view of the flagella. The structure that lies outside the cell wall body, that is the filament portion, here is the hook portion, and this one is the basal body. Now the arrangements of bacterial flagella. If one flagellum is attached to the one pole of the bacterium, it is known as monotrichous flagella, and it is seen in case of Vibrio cholerae. When there is a tuft of flagellum, it is attached to the one pole. It is known as Lophotrichus aciliformis. When there is tuft of flagella situated at both the poles, it is known as Amphitrichus, the third image, and it is seen with the spirillum serpens. And when the flagella is surrounded all around the body, it is known as the Peritrichus flagella, and it is seen in case of Escherichia coli. Now, there is one exception that is seen in the spirochetes. They are motile without any external flagella, but they have one special structure that is known as the axial filament. This axial filament, it is just the bundle of flagellum-like structures that lie between the cell surface and outer sheath, and that connects one end of the cell to the other end. And one structure is the endoflagella. Now we can demonstrate that this bacteria or particular bacteria which are present in the sample of the patient, they are motile or not by microscopic observation in a wet mount preparation, and the other is the hanging drop preparation. Now this wet mount preparation is made like this, in which the slide is taken, and on the center portion you can keep a liquid small portion of the sample, and onto which the cover slip is placed, and this preparation is known as wet mount preparation, that you can see directly under the microscope and evaluate that the bacteria which are present in the sample are motile or not. Another is the hanging drop preparation, that is particularly useful to demonstrate motility of Vibrio cholerae, in which there is concave slide which is taken, and the sample or the liquid stool is placed on the center portion of the cover slip. Then on the edges of cover slip, we can keep a petroleum jelly, and after that we use that concave slide and keep that onto the center portion of the cover slip. Then we can just rotate the whole the preparation, so the center portion of the slide, it will look like a hanging drop preparation, that you will just learn in your practical sessions more clearly. So there are two phenomena that you have to understand. First is the true motility of the bacterium, and the other is the Brownian movement. Now, true motility is itself by the presence of flagella. They can motile by themselves. Okay, so bacteria that swim from one side of the microscopic field to the other side, in the direct microscopic observation, we can say that this bacteria are truly motile and having the flagella in their structure. Now, what is the Brownian movement? It is just due to random collisions between water molecule and the bacterial cells, or the surrounding structure. They just push the bacterial cells, and you assume that this bacteria are motile, but actually they are not motile. Surrounding the bacterial cell, they impart them the motility, that is known as the Brownian movement. Another method is by using semi-solid medium, in which the agar concentration is used is less compared to the solid media, that is around 0.5%, and in which the specimen or the sample is inoculated with the straight line, that is known as a stab culture. Okay, the method of inoculation you will learn in culture media and the inoculation method practicals. So after incubation, the medium is observed for the growth. Here you can see the two test tubes, in which there is a restricted growth along the stab line. So this particular organism is non-motile. And whenever there is diffuse growth around the stab line, we can say that this sample possess the bacteria which are motile. Here is the picture in which the sample is stained with the flagellar stains, and you can appreciate the flagella of the bacterium. Now, functions of flagella. It is responsible for motility of the bacteria, and they are highly antigenic. They constitute a group of protein antigens. They are called as of given species, strain, or variant of an organism. So flagellar antibodies which are produced are not actually protective, but they help in the serodiagnosis because they are characteristic of particular organism only. Now another appendage is fimbriae or pili. They are very hair or thin, like filamentous structure that extend from the cell surface, and they are always peritrichous in arrangement. almost 100 to 500 pili or fimbriae are found in one cell, and they are very short, thin, and more numerous compared to flagella. They are composed of protein subunit pillin, and the flagella is composed of flagellin, and they are also antigenic, just like flagella. Now, types of fimbriae: the common pili or fimbriae, and another is the fertility pili. They are specialized pili, and they are longer and fewer in number compared to common pili, and they are found in male bacteria. So it will helpful for transfer of genetic material by forming a hollow conjugation tubes. Common pili acts as organs of adhesion for the organism. So the bacterial adhesion to the epithelial surfaces help in colonization of particular bacteria by the common pili. Then bacterial adhesion to the red blood cells present in gram negative and some of the gram positive bacteria. Now this fertility pili or the sex pili, they transfer the genetic material from one bacterium to another, and they are present in gram negative bacteria only, just like endotoxin that we have learned in last lecture. So this is the picture, real picture. In this picture or the first left side picture, you can see the dividing shigella, and there are a lot of fimbriae around it. Okay, and in this there is dividing salmonella, in which you can appreciate the long flagella and this small and numerous fimbriae also. The third and very important structure is the spore. They are highly heat resistant, dormant stage of bacteria and formed in unfavorable environmental conditions like starvation and desiccation. Okay, so they form within the parent bacterium and they are known as endospores. Sporulation is the process of formation of endospore, and it is an unusual property of certain bacteria only. Now keep this thing in mind that each bacterium form only one spore, and during germination bacterium. Spore forming bacteria are very specific. They are falls into two categories like obligate aerobic and obligate anaerobic category. In the obligate aerobic category, we found Bacillus anthracis, Bacillus subtilis, and Bacillus cereus. And obligate anaerobic category, that is mainly Clostridium species like Clostridium tetani, Clostridium welchi, and Clostridium botulinum. This is the cross section of a bacterial spore, just like a anthrax, and that shows the hard multi-layered coats, which both makes the spore difficult to kill and allow it to remain in the dormant stage for many years. Okay, so first is the inner membrane, then there is a cortex, then outer one is the coat, and then exosporium. So this is the cycle of spore formation. That is starting from the vegetative cell and it ends into the free endospore, and that again converted into the vegetative cell. This is the cycle of spore formation and germination. So at the beginning of spore formation, the septum is formed that separate the nascent spore from the rest of the cell, and all of the genetic material of the cell is copied into the newly forming cell. The spore contents are dehydrated and the protective outer coating are laid down. And once the spore is matured, it is released from the cell. On germination, the spore contains rehydrate and new bacterium emerges and multiplies. Whenever the environmental conditions are favorable for the germination of the free spore. Now the structure and the arrangement of the spore or the shape of the spore. So spores may form central stage or subterminal stage. These are the categories. Here you can see in the A, the spore shape is oval and it is situated terminally. In the B, the spore shape is rectangular and it is also situated terminally. In the C, same, the shape remains the same, but the situation or the arrangement is subterminal. In the D, it is rectangular in shape and it is central in position. In E, it is circular and terminal in position. In F, circular and central in position. And G, it is terminal, but this cup shape, it is bigger than the size of the cell. Now we can gram stain in our day-to-day practice. Just like the capsule, in the gram stain, this spore they remain unstained. So they are appear as a refractile body within the bacterial cell. In these two picture, if you can appreciate, in which there is a clear area, and that is the spore, that is the central spore. Okay, we can also see them in a modified Zeden stain or Zelen-Nielsen stain as an acid-fast structure. So they appear in the red in color. Uses of the spore. Basically, they are employed as a control checklist, and two spores of bacterium they are most commonly used are the Bacillus stereothermophilus and Bacillus subtilis. This Bacillus stereothermophilus, it is destroyed at the temperature of 121 degrees Celsius in 10 to 20 minutes. And Bacillus subtilis is destroyed at 105 degrees Celsius in 5 minutes. So how they are used in the sterilization process, we keep such indicators due instrument, and after completion of the sterilization process, if you see that this indicator change the color from violet to yellow, then we can say that the spores are actually not killed. And in the process, we see that the spores they germinate and form the vegetative bacteria. So we can certainly say that this sterilization process which we have done, it is not accurate, and we have to repeat the sterilization cycle. So this is all about the morphology of bacterium. In the next lecture, you will learn about the physiology of bacteria that will taken up by the Dr. Yagnesh Pandya. So thank you for today. Have a nice day.